Welcome to the deep dive. Imagine your body not just as a city, but as a vast, constantly evolving metropolis. Every single cell in that city holds the master blueprint. It's DNA. And today we're taking a deep dive into how that blueprint is not only copied with mind-boggling precision, but also tirelessly guarded, patched up, and sometimes even deliberately rewritten.
Right.
We're talking about the very core of life itself: DNA synthesis, repair, and genetic rearrangements.
And what's truly remarkable about this topic, especially as we explore it through the lens of Marks Basic Medical Biochemistry, is how these fundamental biochemical processes are just inextricably linked to clinical realities.
Yeah.
This isn't just about molecules floating around. It's about understanding the why it matters.
Right.
You know, why we get bacterial infections, how certain cancers arise, and honestly, how many of our most effective medicines actually work.
Exactly.
We'll guide you step by step through these concepts, making sure we distill those crucial insights for you.
Absolutely. Think of it as peeling back the layers to see the molecular machines that keep us going, what happens when they encounter a challenge, and, well, the surprising ways life adapts. We've got some fantastic examples from our source material that really bring this science to life. So let's pull back the curtain on the most fundamental process first: DNA replication. Before a cell can divide and create a new cell for our body's growth or repair, it has to copy its entire genetic library. How does it manage that immense task without, making a complete mess?
It's actually a remarkably elegant process. It's called semiconservative replication.
Conservative. Okay.
Yeah. Picture the original DNA double helix. That's our master blueprint, right? During replication, those two strands separate, and each one acts as a template for building a brand new complementary partner strand.
Okay.
So every new DNA molecule you end up with is a kind of hybrid, one old parent strand and one freshly synthesized strand.
Like keeping half the old instructions and adding a perfect new half.
Exactly. It's like keeping one half of the original instruction manual and binding it with a perfectly matched, newly printed half. This mechanism ensures incredible fidelity in passing on genetic information.
That makes a lot of sense. You always have a reliable guide for the new copy. But how does this copying frenzy actually begin? Where does it kick off?
It all starts at specific points, opening up what we call a replication fork.
Replication fork.
Yeah, think of it literally as a zipper unzipping the double helix. As this fork moves along the DNA, it forces the two parental strands apart. And right behind it, the assembly lines are busy building those new strands. It takes a whole complex of proteins and enzymes orchestrating this. It's quite amazing.
A molecular construction crew. And who are the key players in getting that zipper unwound and making sure those separated strands don't just snap back together? Especially in, say, a simpler system like a bacterium, like E. coli.
Right, the prokaryotic model. Our source highlights several crucial enzymes that act as the opening crew. First, you have helicases.
Helicases.
Yeah, like the Dianne A protein in E. coli. These are the actual unwinding enzymes, like tiny, powerful motors just forcing the strands apart.
Okay.
And as the DNA separates, single-strand binding proteins immediately jump onto the exposed strands. Their job is basically to keep those strands apart, preventing them from reforming the helix until they're copied.
Stabilizers.
Exactly. And then there's a really ingenious solution to a problem that arises from all this unwinding: topoisomerases.
Topoisomerases. What do they do?
Well, imagine twisting a rubber band. The more you twist one end, the more it coils up further down, right?
Yeah, it gets super tight.
That's what happens to DNA ahead of the replication fork. It creates immense tension, what we call supercoiling. Topoisomerases are like molecular mechanics. They relieve this tension. They actually cut one or both DNA strands, allow the tension to unwind, and then they rejoin them.
Wow. Snip, unwind, reseal.
Yeah.
That's clever. And I imagine this makes topoisomerases, especially the bacterial ones, a prime target for medicine.
Absolutely. And here's where it gets clinically fascinating, Fano. In bacterial cells, a key topoisomerase is called DNA gyrase.
DNA gyrase.
And drugs like ciprofloxacin, it's a common quinolone antibiotic, are specifically designed to inhibit bacterial DNA gyrase.
Okay.
By stopping this critical unwinding process, they halt bacterial DNA synthesis, effectively treating bacterial infections. We see this in cases like Dianne A's urinary tract infection mentioned in the source, which responds well to ciprofloxacin.
And crucially, they don't mess with our own cells.
Precisely. Human cells use different topoisomerases, so these drugs don't harm us, making them effective and, relatively safe.
Brilliant. So the DNA zipper is open, it's stable, the tension's managed, the stage is set. But what's the actual engine of this copying process? Who's laying down the new genetic bricks?
That's the monumental task of the DNA polymerases. In E. coli, Pol I is the primary enzyme doing the heavy lifting of replication.
Both are.
Now, all DNA polymerases are very particular. They read the original template strand in one specific direction, the 3 to 5 direction, and they can only build the new strand in the opposite, 5 to 3 direction.
Always 5' to 3' for building.
Always. Imagine a train that can only lay track going forward, even though it's following a track laid in reverse. The raw materials for these new strands are deoxyribonucleoside triphosphates, DNTPs.
Right, the building block.
Yes, and they're like individual building blocks with their own built-in energy packs. The energy for adding each new nucleotide comes from cleaving off 2 phosphates pyrophosphate, releasing a burst of power.
Energy included. Efficient.
Very, and these polymerases are also incredibly processive.
Processive, what does that mean?
It means they stay clamped onto the DNA template for long stretches, sometimes thousands of bases, without falling off. This allows for incredibly fast and efficient synthesis.
Makes sense. But can DNA polymerase just start building from scratch, anywhere on the template? It sounds like such a precise operation, it must need a clear starting signal.
You're absolutely right, it can't. Every DNA polymerase needs a sort of launch pad, a primer.
A primer, like painting a wall.
Exactly like that. And this primer isn't DNA, it's actually a short RNA oligonucleotide, a temporary starter segment. It's synthesized by an enzyme called Primase.
So RNA starts the DNA chain. Interesting.
It is. The DNA polymerase then latches onto the 3' hydroxyl end of this RNA primer and starts adding the DNA building blocks, the deoxyribonucleotides, initiating the new DNA strand, like having that small pre-built foundation before you can start constructing the main wall.
So, okay, if the polymerase can only build in that one specific 5' to 3' direction,
Yeah.
and the 2 DNA strands run in opposite ways anti-parallel,
Or anti-parallel.
how on earth do both sides of that unzipped helix get copied at the same time? That sounds like a fundamental design puzzle.
It absolutely was a puzzle, and it was solved beautifully by Okazaki's discovery. It really boils down to 2 different strategies for the 2 strands. On one template strand, the leading strand, synthesis is continuous. The polymerase just zips along smoothly, following right behind the replication fork as it opens.
Simple enough.
But on the other strand, the lagging strand, because of that strict 5 to 3 foot synthesis rule and the anti-parallel nature, the polymerase has to work backwards in short bursts. It has to wait for the fork to open up a stretch, then synthesize back away from the fork.
Ah, okay, so it's discontinuous.
Exactly. This creates many small, disconnected segments known as Okazaki fragments, which are synthesized away from the direction the replication fork is moving. Imagine one continuous highway being built and another road built section by section, like short, unconnected on-ramps.
Okay, so you have all these little Okazaki fragments on the lagging strand, each started with an RNA primer. How do they get stitched together into a complete seamless strand? There must be a molecular cleanup crew involved.
Precisely. It's a multi-step cleanup operation. First, specialized enzymes like RNase H and another DNA polymerase I, Pol I, in E. coli, work together to remove those temporary RNA primers.
Get rid of the starter blocks.
Right. Then, Pol I steps in to fill in the resulting gaps with actual DNA nucleotides.
Okay.
And finally, an enzyme called DNA ligase acts as the molecular glue. It seals all the tiny nicks between the fragments, joining the Okazaki fragments together to form one continuous, flawless strand.
Ligase the stitcher. Got it.
Now, beyond just building, DNA replication boasts incredible accuracy. Pol I in E. coli, for instance, has a built-in proofreading function. It's essentially a molecular delete key.
A spell checker for DNA?
Kind of. It's a 3 to 5 exonuclease activity. This means if it accidentally adds an incorrect nucleotide, it can literally back up one step, recognize the mismatch, snip out the wrong base, and then replace it with the correct one before continuing.
Wow.
This proofreading reduces the error rate dramatically, down to about 1 mismatch in a million bases incorporated. And when you factor in other backup repair systems, the overall error rate can be astonishingly low, maybe 1 in 10 billion.
That's an absolutely astonishing level of precision. And I'm guessing this proofreading ability, or maybe the lack thereof, has critical implications, especially when we think about viruses.
It absolutely does. This is a crucial insight from our source material. For instance, HIV's reverse transcriptase, that's the enzyme it uses to copy its RNA genome into DNA, it lacks this robust proofreading activity.
Ah, no spell checker for HIV.
Exactly. And this is a major reason why HIV mutates so rapidly. The virus is constantly making errors, constantly evolving. This makes it incredibly challenging to treat, as we see with Isabel S's complex multi-drug regimen mentioned in the text. The constant mutations allow it to develop resistance to drugs pretty quickly, forcing continuous adaptation in treatment strategies.
Okay, that bacterial replication is incredibly efficient, a real masterclass in copying. But now imagine scaling that challenge up thousands of times linear chromosomes, vastly more DNA, a finely tuned cell cycle.
Yeah.
Our human cells, eukaryotic cells,
Yeah.
face even greater hurdles, right?
Yeah.
They also have evolved some unique solutions.
Absolutely. While the core machinery, helicases, polymerases, ligases remain similar, eukaryotic cells introduce layers of complexity that reflect their much larger genomes and the linear nature of their chromosomes, all tucked neatly within the nucleus.
So if we connect this to the bigger picture, where does this massive DNA copying effort fit into the entire life story of a human cell? Is it happening all the time?
No, it's very tightly orchestrated within what we call the cell cycle. DNA replication specifically happens during the S phase or synthesis phase.
For synthesis, okay.
Right. The cell cycle is like a precisely timed molecular clock. It moves through different phases. G1, that's the first gap. S synthesis, G2, second gap, and finally M, mitosis or cell division.
G1S, G2M.
Correct. And cells can also enter a quiescent sort of resting state called G0. Think of our mature liver cells, they're often in G0.
Saying out, not dividing.
Exactly. But even these can be stimulated to re-enter the cycle, divide, and regenerate. Our source actually mentions the ancient myth of Prometheus.
Whose liver was eaten by an eagle and regenerated daily.
Right, which, strikingly mirrors this known capacity of our liver cells to jump back into action from that quiescent G0 state and regenerate tissue. It's pretty cool.
It's incredible how some ancient stories seem to tap into these profound biological truths, even without the molecular details. So this ability for certain cells to essentially pause and then restart division, that's a powerful strategy.
Indeed, it speaks to our body's remarkable capacity for repair and renewal. Now, you asked about scale. How do we replicate all that human DNA efficiently, especially with our massive linear chromosomes?
Yeah, it seems like it would take forever with just one starting point.
It would. So the key here is efficiency through multiplicity.
Mm.
Instead of a single origin point like in E. coli, eukaryotic chromosomes have multiple points of origin scattered all along their length.
Many starting lines.
Exactly. This creates many bubbles of replication forks that initiate simultaneously, enlarge, and eventually merge together. It's like having hundreds of construction crews starting work at the same time across a vast city, allowing rapid duplication of our large chromosomes within just a few hours during that S phase.
Makes sense. And do we use the same polymerases as bacteria, or is it a different team?
It's a much more diverse team. We have around 15 different DNA polymerases in eukaryotes.
15, wow.
Yeah. The main workhorses, the major replicative enzymes, are Pol, delta, and Pol epsilon. They play roles similar to Pol third back in bacteria.
Okay, delta and epsilon.
Then there's Pol alpha.
Oh.
It's crucial for initiating synthesis. It actually works together with Primase to produce that RNA primer, and then adds a short stretch of DNA to get things started.
Reinitiator.
Right. And interestingly, there's Pol gamma, which is found exclusively in mitochondria.
Mitochondria have their own DNA polymerase.
They do. And it highlights the sort of bacterial-like origin and nature of mitochondrial DNA replication. We even have specialized bypass polymerases that can kind of muscle their way past damaged DNA regions, ensuring replication can continue even when the template isn't perfect, although sometimes that comes at the cost of making more errors.
A complex toolkit for a complex job.
Mm-hmm.
How does the lagging strand synthesis work with all these players?
It's a highly coordinated handoff. Pol I and its associated Primase laid down those initial RNA primers for the Okazaki fragments on the lagging strand. Then, after Pol I detaches, Pol I takes over to extend the fragment with DNA.
A switcheroo.
Pretty much. Subsequently, a series of helper enzymes like FEN1 and RNase H come in to remove the RNA primers. Pol I fills the gap, and then DNA ligase acts as the final molecular stitcher, sealing everything up.
And the Okazaki fragments themselves.
Interestingly, you'll notice that eukaryotic Okazaki fragments are much smaller than in prokaryotes, often corresponding pretty closely to the length of DNA wrapped around a single nucleosome. Those protein spools DNA is wound around.
Related to the chromatin structure. And what about ensuring this whole complex process is organized and doesn't just fall apart? keeping those polymerases on track.
A critical protein, often the unsung hero in diagrams, is pCNA, that stands for proliferating cell nuclear antigen.
pCNA.
It acts like a sliding clamp. It forms a ring around the DNA and holds the DNA polymerases, particularly Pol and Ne, tightly to the template strand. This greatly increases their processivity, making sure they don't fall off easily.
So it keeps the machinery locked on.
Exactly. And pCNA is so indicative of actively dividing cells that it's even used clinically as a marker for cell proliferation, for example, in assessing tumors in cancer diagnosis.
Very useful. Okay, now here's where it gets really fascinating for me. Human chromosomes are linear, right? Not circles like bacteria. What happens when replication reaches the very, very end? It feels like there's a fundamental problem with that 5-to-3 synthesis rule on the lagging strand.
You've hit on a major challenge, spot on. It's known as the end replication problem.
And replication problem.
Yeah. On the lagging strand, when you remove that very last RNA primer at the extreme 5-foot end of the newly synthesized strand, there's no upstream primer for DNA polymerase to extend from to fill the gap.
So you're left with a gap, a shorter end.
Exactly. You're left with a shortened 5-foot end and a corresponding single-stranded 3-foot overhang on the template strand. Without a special solution, our chromosomes would indeed get progressively shorter with each round of replication, eventually losing vital genetic information from the ends.
That sounds bad, like planned obsolescence for our cells. How do we fix it?
This is where a remarkable enzyme called telomerase comes to the rescue.
Telomerase. I've heard of this.
It's a specialized enzyme, technically an RNA-dependent DNA polymerase, also called a reverse transcriptase, interestingly enough.
It makes DNA from RNA.
Yes, because it carries its own internal RNA template within the enzyme complex. In humans, this RNA template has a sequence complementary to our telomeres repeating sequence, which is TTTG over and over again.
TTG repeats at the ends.
Correct. So telomerase binds to that single-stranded 3' of overhang left by replication. It then uses its own internal RNA as a template to extend that 3' end, adding more TTG repeats.
It lengthens the overhang.
Precisely. This lengthening creates enough space further down for Primase to come in, synthesize a new RNA primer, and allow DNA polymerase to then fully complete the lagging strand synthesis, effectively preventing the chromosome from getting shorter.
So it tops up the ends. But doesn't this relate to aging in cancer?
Absolutely. This telomerase mechanism raises profound questions. Why do some cells seem to live forever while others age and die? Telomerase activity is definitely linked to cell aging in cancer. Most of our normal body cells, somatic cells, don't express much telomerase.
Oh, their telomeres shorten over time.
Yes, leading to limited cell divisions, a state called senescence. It's like a cellular clock. However, stem cells, which need to divide extensively, and unfortunately most cancer cells do express telomerase, this allows them to maintain telomere length and achieve a sort of cellular immortality.
So this telomerase paradox isn't just about cell longevity. It reveals a profound evolutionary balancing act. For most cells, limited telomerase acts as a built-in tumor suppressor, preventing runaway growth.
That's a great way to put it.
But for vital stem cells, that immortality is a must for healing and renewal, making telomerase this double-edged sword, a key to both life's regeneration and its vulnerabilities. Like cancer. That's a powerful insight. So DNA replication is this incredibly precise operation, with ingenious solutions to its inherent challenges. But even the best systems have flaws, and our DNA is under constant attack from the environment. What happens when errors slip through that proofreading net, or when DNA gets damaged by outside forces?
Yeah, despite that remarkable proofreading, some mismatches will inevitably persist. But honestly, a far greater threat often comes from mutagens.
Mutagens, things that cause mutations.
Exactly. Environmental chemicals or various forms of radiation that physically damage the DNA, leading to mutations if not repaired. And mutagens that specifically cause cancer are called carcinogens. The reality is our DNA faces thousands of potentially mutagenic lesions, hits, damages every single day.
Thousands a day. Wow.
It's constant. Without a robust and tireless cellular repair crew working around the clock, we simply couldn't survive these constant assaults.
What kind of damage are we talking about? What are the common culprits?
Well, our source details several key examples. Radiation, for instance, like X-rays or even background radiation, can generate highly reactive oxygen species, think hydroxyl radicals inside the cell. These can directly damage the DNA bases or even cause breaks in the DNA strands.
Okay, radiation's one.
Then there are chemicals. Many carcinogens, like benzopyrene, which is found in tobacco smoke and coal tar, A factor in Michael T's lung cancer case from the text can react with DNA bases, particularly guanine. They form bulky attachments or adducts.
Bulky adducts, they distort the helix.
Exactly. They physically distort the DNA double helix, interfering with replication and transcription. And of course, there's UV light from the sun. This is the likely cause of Calvin A's melanoma in the source material. UV rays primarily excite adjacent pyrimidine bases in the DNA, especially thymines, causing them to covalently bond together and form thymine dimers. These also distort the helix significantly.
Sunburn is literally DNA damage. Okay, it sounds like our DNA is constantly under siege. Fortunately, our cells aren't just sitting ducks, right? They must have ways to fight back to patch things up.
Oh, absolutely. Our cells possess an astonishing arsenal of DNA repair systems. It's quite complex, but most of them follow a general kind of logical four-step scheme.
Okay, what's the basic plan?
First, the cell has to recognize that there's damage or a distortion in the helix. Second, enzymes remove the damaged section, often cutting out a piece of the affected strand.
Recognize and remove.
Third, a DNA polymerase steps in. It uses the intact complementary strand as a pristine template to fill the gap with the correct nucleotides. And finally, step 4, DNA ligase performs its usual molecular glue job, sealing the final nick in the backbone to restore the intact double helix.
Recognize, remove, replace, reseal. Got it. Are there different systems for different types of damage?
Yes, very much so. Let's touch on a few key pathways mentioned in the source. Nucleotide excision repair, NER, is a major one. This system is designed to handle larger helix-distorting lesions, like those bulky adducts from carcinogens or the UV-induced thymine dimers we just discussed.
So NER tackles the big stuff.
Right. Specialized enzymes recognize the distortion, make cuts on either side of the damage, and remove that entire abnormal segment, which might be 20-30 nucleotides long. Then, a DNA polymerase fills the gap using the other strand as a template, and ligase seals it.
And this leads directly to a very serious clinical connection, doesn't it, if NER fails?
It absolutely does. Defects in the NER pathway cause a devastating genetic disorder called xeroderma pigmentosum, XP. Individuals with XP are extraordinarily sensitive to sunlight. They develop severe sunburns very easily and have a massively increased risk, like thousands of times higher, of developing skin cancers, often at a very young age. It's precisely because their cells can't effectively repair the UV-induced DNA damage, leading to a dangerous accumulation of mutations over time.
A stark example of why repair is so vital. What about smaller damages?
For smaller single-base distortions, there's base excision repair, BER. This system targets issues like when a base gets chemically modified, for instance, when cytosine spontaneously deminates, loses an amino group, and turns into uracil. Uracil doesn't belong in DNA.
Right, used usually in RNA.
Exactly. So a class of enzymes called DNA glycosylases specifically recognizes and removes just the damaged base itself, leaving the sugar phosphate backbone intact but creating a gap called an AP site, a pyrenic site. Other enzymes then process this AP site, remove the sugar phosphate remnant, a polymerase inserts the correct base, and ligase seals it up. Without BER, that CG pair could incorrectly become a TA pair after replication, leading to a permanent point mutation.
So BER handles the small chemical tweaks. What about simple typos from replication that proofreading missed?
Good question. That's where mismatch repair, MMR, comes in. Even with proofreading, occasionally a normal base gets incorrectly paired during replication, say, an A accidentally paired with a C instead of a T. Mismatch repair enzymes can detect these non-Watson-Crick pairings after replication is finished.
How do they know which strand is the original correct one and which is the new one with the error?
That's the clever part, especially in bacteria. They distinguish the old template strand from the newly synthesized strand by methylation patterns. The template strand is methylated shortly after replication, but the new strand isn't immediately methylated. So the MMR system knows to fix the error on the unmethylated new strand. Human cells have similar sophisticated MMR systems, though the exact mechanism for strand discrimination is still being fully worked out.
And again, I sense a critical clinical link here, particularly to cancer, if MMR fails.
Indeed. Mutations in mismatch repair genes are directly linked to a common form of hereditary colon cancer called hereditary non-polyposis colorectal cancer, HNPCC, also known as Lynch syndrome.
HNPCC or Lynch syndrome.
Right. Individuals with this condition have a significantly increased lifetime risk of colorectal cancer, as well as other cancers like endometrial cancer. The inability to correct these simple replication mismatches allows mutations to accumulate much more rapidly than normal, especially in genes that regulate cell growth, ultimately driving cancer development.
Fascinating and scary. Are there other specialized repair systems?
Yes, a couple more highlighted. There's transcription-coupled repair, TCR. What's interesting here is that genes that are actively being transcribed, actively making RNA copies, seem to get preferential treatment for repair.
Priority service for active genes.
Kind of. If an RNA polymerase, while transcribing a gene, physically stalls at a site of DNA damage, it acts as a signal. Repair proteins are recruited directly to that spot to fix the DNA right there, allowing transcription to resume quickly. It makes sense to prioritize fixing the genes currently needed by the cell. Defects in this TCR system can lead to rare genetic disorders like Cockayne syndrome, characterized by severe developmental problems, premature aging, and neurological issues.
Okay.
And perhaps connecting this to the bigger picture of human health and disease again, many hereditary cancers, including those linked to the well-known BRCA1 and BRCA2 mutations.
Associated with hereditary breast and ovarian cancer risk.
Exactly. These are often due to defects in broader DNA repair proteins, particularly those involved in fixing the most dangerous type of damage. Double-strand breaks where both strands of the helix are severed. Repairing those accurately is critical to maintaining genomic stability.
So a whole suite of repair systems constantly patrolling and fixing. But while most of these mechanisms are about fixing errors or damage, you mentioned DNA can also deliberately rearrange itself. What's the purpose of that? It sounds a bit chaotic for something so fundamental.
It's a powerful point. Genetic change isn't always detrimental. In fact, it's the engine of evolution. But you're right, these rearrangements can also have devastating consequences. One mostly beneficial example is homologous recombination.
Homologous recombination.
This involves the physical exchange of DNA segments between two very similar or homologous DNA molecules. You see this most famously during meiosis, the process that makes sperm and eggs, where it's called crossing over. It shuffles genes between parental chromosomes, increasing genetic diversity in offspring. Homologous recombination is also a vital pathway for accurately repairing those dangerous double-strand breaks we just mentioned, using the undamaged sister chromatid as a template.
So that's generally a good thing. What about problematic rearrangements?
Well, sometimes chromosome breaks happen and the repair process goes wrong. This can lead to translocations, where a piece of one chromosome breaks off and mistakenly gets joined to a completely different chromosome. These gross chromosomal rearrangements are frequently observed in cancer cells. They can activate oncogenes, cancer-causing genes, by moving them to a highly active region, or they can disrupt tumor suppressor genes, contributing to uncontrolled cell growth.
So accidental, large-scale reshuffling.
Exactly. And then there's another fascinating category, transposable elements, which were famously discovered by Barbara McClintock and initially dubbed jumping genes.
Jumping genes, they move around.
They literally can.
Yeah.
These are specific segments of DNA that have the ability to move from one location in the genome to another. Some just cut themselves out and paste elsewhere. Others, called retroposins, work through an RNA intermediate. They get transcribed into RNA, then use reverse transcriptase, that enzyme we discussed with HIV, to make a DNA copy of the RNA. And that DNA copy then gets inserted into a new location in the genome. These elements make up a surprisingly large fraction of our genome, and while much of it is inactive, their movement can sometimes cause mutations or alter gene regulation.
We touched on reverse transcriptase with HIV and just now with retroposins. Let's circle back to that. What exactly is this enzyme, and why is it such a unique player? It seems to go against the central dogma, the fundamental flow of genetic information.
That's its defining characteristic, really. Most of the time, as you say, genetic information flows from DNA to RNA transcription, then RNA to protein translation. That's the central dogma. But reverse transcriptase completely breaks that rule. It does the reverse of transcription.
Makes DNA from RNA.
Exactly. It uses a single-stranded RNA template to synthesize a DNA copy. We call this DNA copy complementary DNA, or cDNA. It's literally reversing the usual direction of genetic information flow. RNA back to DNA.
So it's essentially working backwards in the usual genetic flow. And what does this mean clinically? We know HIV uses it.
Right. This enzyme is primarily found in and essential for retroviruses like HIV. It's a core part of their life cycle. The virus particle contains an RNA genome, and upon entering a host cell, It uses its reverse transcriptase to convert that RNA genome into a double-stranded DNA copy.
And then what happens to that viral DNA?
That neurally synthesized viral DNA can then integrate, basically insert itself, into the human host cell's genome, becoming a permanent part of our genetic material in that infected cell.
Wow, it becomes part of us.
Effectively, yes, in those cells. This integration is what allows the virus to persist and replicate, potentially leading to diseases like AIDS. In some cases, integration near cellular genes involved in growth control could even contribute to certain cancers, though that's more common with other retroviruses than HIV itself.
So targeting this unique enzyme is key for HIV treatment.
Absolutely critical. This is why drugs that inhibit reverse transcriptase, like lamivudine used by Isabel S in the clinical case, are such cornerstones of HIV therapy. They specifically block this essential viral DNA synthesis step, preventing the virus from establishing that permanent infection by embedding itself in our cells. It really highlights the power of understanding these unique molecular pathways for designing effective therapeutic interventions.
Wow, what a journey we've taken. From the precise, coordinated dance of those replication forks to the tireless, vigilant work of our cellular repair enzymes, and then the surprising flexibility offered by genetic rearrangement. Our DNA truly is a marvel of biological engineering. So for you, our listener, thinking about all this complexity, what's the most crucial takeaway from this intricate process described in Marx's?
Yeah, it's a lot to take in, but if we connect it all back to the bigger picture, I think you've really seen today how understanding DNA synthesis and repair isn't just some academic curiosity. It's absolutely fundamental.
Fundamental to what, specifically?
Fundamental to comprehending how our cells divide and tissues grow, how devastating diseases like cancer and HIV develop their insidious strategies, and crucially, precisely how a vast array of our modern medicines actually achieve their therapeutic effects by targeting these very pathways. The molecular mechanisms we've discussed, they're under constant assault, both from internal errors and external damage. Their fidelity, their ability to copy accurately and repair effectively, is directly linked to our health, our survival, and even the process of how we age. This chapter from Marks Basic Medical Biochemistry truly lays the bedrock for so many vital clinical insights.
It really does make you appreciate the constant, invisible work happening inside us every second. The sheer ingenuity of life itself is just staggering. And this delicate balance you describe between absolutely precise copying for stability and yet allowing for some capacity for strategic change or repair, it's what allows for both biological continuity and the incredible dynamism of evolution.
Well said.
It also makes you wonder, doesn't it? With our growing ability to actually manipulate these very processes, think gene editing technologies like CRISPR, what are the next frontiers in medicine? And perhaps just as importantly, what new ethical questions or considerations about life itself might arise as we push those boundaries further?
That's the million-dollar question, isn't it? Huge potential, but also huge responsibility.
Indeed. Thank you so much for joining us on this deep dive into the absolutely fascinating world of DNA synthesis, repair, and genetic change. We really hope this has given you some great insights from the source material and perhaps even sparked some new questions for you to explore on your own.